Good evening. This is Shane Gebauer with BrushyMtnBeeFarm. I'm the general manager. This evening, as you I'm sure are well aware, we've got Dr. David Tarpy with us from the Department of Entomology at North Carolina State University. Dave, I'd like to thank you for coming this evening. He is, for those of you who may not recognize the name Dr. David Tarpy, he is one of the people that I often refer to when I'm talking about queen quality, queens that may be superseded in sort of the quality of those queens. He's done a lot of really great research, and he's recognized as one of the key figures in queen rearing and research going on in that area, looking at mating behavior and reproductive success, competition between queens, and things like that. He's also doing some work with CCD and how nutrition may be playing a role in that, as well as the genetics of a queen. And so we really are quite fortunate to pin him down for the next hour or so and hear about some of the things he's doing. And so, Dave, I'd like to thank you for coming. I really do appreciate you taking the time to be with us this evening, and I'm sure we're going to benefit greatly from what you've got to discuss with us this evening. So I'll turn it over to you.
It's my pleasure.
All right.
Okay.
It's all yours.
Okay, just let me know when you're going to record.
It's all set.
Okay, all set. Well,
yep.
Thank you, Shane, for the invitation, and thank you, everybody, for attending. This is kind of a new format for a lot of us, or at least me certainly, but I definitely see the power and the draw that these type of webinars may have for discussions and for the apiculture community to really come together and discuss issues that otherwise might be more difficult to do in travel and in other sort of formats. And so I really applaud Shane and Brushy Mountain for really being on the forefront of this type of technology, and I definitely think that we're going to see more things like this in the future, and it's gonna really benefit all of us. So when Shane approached me about doing a webinar, he requested that one of the topics that may be of interest to you listeners out there may be on some of our recent research on commercial queens, and in particular the quality of their mating. As we'll talk about for the next hour or so, I think there may be some real issues here dealing with the queens in our hives, and how well mated they are may be indicative of how good the colony is. And so we'll kind of explore some of the research that we've been doing in this particular area. As Shane mentioned briefly earlier, a lot of our research previously in our lab has really dealt with the benefits of queens and the benefits of queens mating multiply and the consequential genetic diversity among the worker force within colonies. And so a natural extension of a lot of that work was to really see what the mating quality of commercial queens are. So before I begin, I just need to, of course, up front mention all of the really excellent folks that have contributed to the research that I'll be presenting today. So I would be neglectful to think that this is just a singular effort, by no means. We have really excellent field technical help with Jennifer Keller, who's our apiculture technician out in the field. So anything really dealing with live bees, she was really spearheading and really does a tremendous job for us here at NC State. Her counterpart in the genetics laboratory is Joel Karen. Joel is really responsible for organizing and spearheading all the genetic analyses and some of the more kind of cutting-edge technological assays that we've done. A third very key member of our team here is Dr. Deborah Delaney, who is a postdoctoral researcher in our lab who really brought in a lot of the skill sets specifically some of the genetic work and some of the sperm staining work to really try to understand the mating quality of queens. And then we've had several undergraduate or student researchers that helped analyze the data that I'll be presenting, and so we really thank them for their efforts as well. And of course, the work that we'll be presenting is by no means cheap. It takes quite a lot of personnel time as well as financial resources to accomplish things like this. And so I also want to recognize many of the groups that provided funding for this research, specifically the USDA with a very large grant from the USDA entomology program, but as not as large grants, but certainly very, very appreciated and helpful from the North Carolina state beekeepers, the California state beekeepers, as well as the North Carolina Department of Agriculture. So with that,
Dave, I just want to, if I may, I'm sorry
yes.
to interrupt, but I just want to do one quick technology check here. Someone's posted in that they don't have sound, so I'm not sure if it's simply this one person. If someone else out there can hear us, I'm seeing that everything's working correctly, but could someone just let us know if they hear us okay? I see a hand coming up, which suggests that we've got, I see lots of people raising their hands suggesting that they've got sound. So I'm sorry to interrupt you there, Dave. I just wanted to make sure that we didn't have some problem there with our sound.
Yep.
But it seems like it's working just fine. So
okay,
go
problem.
ahead.
Although it's very possible that after the next hour you're going to wish that I was on mute the whole time. But,
I'm sure that won't be the case.
All right. Well, hopefully most folks can hear, and so I'll just continue. I've only personally been keeping bees for about 15, 16 years, which for many of you out there that may not be a very substantial period of time. But even within that short period of time, I've noticed that there has been some problems with queens that beekeepers have been experiencing, and they're kind of subtle and diffuse and kind of difficult to describe, and as such there isn't a lot of real kind of good empirical evidence to suggest that something's going on. But I think that innate beekeeper sense that a lot of us have would inform us that there may be some issues going on with the queens. And in fact, some of the best evidence to this fact was a survey done by Dennis Van Engelsdorp and others, who surveyed beekeepers all across the nation. And among many of the things that they asked them of the colonies that died this past winter, what do you as the beekeeper attribute the colony losses to? And the responses came back that about 9% of the colonies were suspected to have died from CCD or CCD-like symptoms. The more commonly, however, was that varroa mites seemed to account for about 24% of the colony losses from the beekeepers. Even more, 28% of the colonies, the beekeepers blamed starvation or some sort of nutritional issue. And so clearly, while CCD is making all the headlines, there are lots of other factors that beekeepers are concerned with as far as the ill health of their colonies. But all of these three issues were still behind the #1 issue that beekeepers self-identified as being problematic, and that was, quote-unquote, poor queens. That for some reason or another, the colony's queens were somehow to blame for their eventual mortality. Sometimes it was the colony tried to requeen and they failed to requeen. Sometimes the queens just simply turned into drone layers. Other times they had very poor shot brood. Other times they just kind of quit laying altogether. So there's a lot of questions and a lot of blame on poor queens being problems for our colonies. And what I'd like to convince you all of is that these issues, or many of these issues, identified by beekeepers, defining them as poor queens, can really be traced to or be attributed to not the queen quality per se, but the queen's mates. In other words, who she's mating with and how many times she's mating. And so that's really kind of the basis of this research, trying to look into some of these issues currently with queens, and specifically with poor queens. So if we take the approach that looking at the queen and making sure that the queen's quality, so that queen's reproductive potential is at its very highest, that will then eventually translate into making the colony as productive and of highest quality. So the more things we can do to ensure that we have high quality queens will go a long way to help us ensure that we have high quality colonies. And so it's not a very easy thing to try to measure queen quality. That is, how does one go about actually defining what makes a good queen and what makes a bad queen? And I think many of you kind of may recognize that this isn't just one singular measurement that you can take, that queen quality really varies and can be measured in many, many different ways. For example, you can look at the physical quality of queens. You can measure her body size, how large she is or how small she is, and other physical measures, and also the degree of parasitism. In other words, does she have parasites that may lower her overall quality. There's also, as I said before, the other aspect of the queen is not just her, but who she's mated with. And so understanding the insemination quality of a queen is really important, both in terms of how much stored sperm she has in her spermatheca, as well as the viability of that sperm. So if a queen is inseminated with, say, 5 million sperm, but for some reason or another all of those sperm are dead or otherwise inviable, then that doesn't really help the queen at all, right? A third aspect is not just her insemination quality and how much sperm she has, but the number of mates by the queen. In other words, her mating quality. So it's not just a function of how much sperm she has, but rather the diversity of sperm from the different drones with which she mated. And so, as I mentioned before, that being a basis of a lot of our previous research, this is an important factor that also is very crucial in trying to understand a queen's overall quality. So knowing that, we went back into the scientific literature, and we looked at similar studies where they investigated various measures of queen quality. So we found, even dating back all the way to 1947, we found studies on commercially produced honeybee queens where they measured these various aspects of queen quality. In this case, their ovary development, their parasitism for both nosema or tracheomites, their sperm counts, and then finally mating number. And so we found throughout the years several studies that have looked at one or more of these aspects of queen quality. And so I'll refer back to this table periodically just to give us kind of a historical perspective, and hopefully that if we see, if we measure what queen quality looks like today, we'll be able to use that historical perspective to see if that particular measure of queen quality has gone up or gone down. So our experimental procedure was actually fairly straightforward, or at least the design, experimental design, was very readily accomplished. What we did is we just simply went to the back of beekeeping periodicals, and we found advertisements for honeybee queens produced commercially that any one of us as beekeepers would buy. And we went to many different producers, I believe it was 12 different producers, and we ordered 12 queens from each. And we didn't do it randomly. We kind of cherry-picked in different parts of the country and small and large producers. So we tried to get a real kind of broad cross-section of the available queens that were out there. I should mention that all of these queens were of Italian stock, so we didn't actually vary the different stocks across queens. So we standardized them by ordering 12 Italian queens from 12 different queen producers. Upon arrival then, we measured them for all of the things that we could think we could measure them for, their different physical, insemination, and mating qualities, as I alluded to earlier. And so the intent of all of this was to answer this question right here: what are we buying? So this was a very kind of beekeeper-centric type of approach where we just wanted to ascertain what one would expect to receive if one were to purchase a commercially produced queen. And in fact, we did this very cryptically, where rather than put the purchase orders through NC State University, where the beekeepers may perhaps cherry pick some of their better queens and send to us just because they might think it's part of a research program. What we did is we had a member of our research team, Joe Flowers, purchase it with his own personal credit card and have it shipped to his apartment address. So it wasn't really involved with NC State at all. So these queens were literally purchased by Joe the beekeeper, just to avoid any sort of bias in that way. So that's in essence what we're asking and trying to answer is, what are we buying as beekeepers from these commercial queens? So the first thing that we did was we measured the queens, the adult queens, for their physical quality. And most specifically, we measured them for very standard measurements of adult insects. So things like their wet weight, where we chilled them for about 3 or 4 minutes in a freezer just so that they would be immobilized but not killed, and then we would place them and weigh them to the nearest tenth of a milligram. We then, using digital calipers, we also measured their thorax width, so kind of the place where their wings go into their thorax, that width of how wide the queens are, not just how heavy they are. Similarly, their head length, and then how long their wings were, both the right and the left wing. And so this plot right here shows the correlations of all of these different factors. And as you might imagine, all measures were very highly correlated, so that wider queens tended to weigh more and vice versa. So that's not all that surprising, but it just gives us an idea of the spread and the differences among queens that we purchased. For example, just looking at wet weight of the queens by the different source colony or source beekeeping operations from which we ordered these queens, there were significant differences within and among these different producers. So these are coded by letters so that to maintain blindness of these different beekeepers. But overall, what we found was that these mated queens weighed an average of about 194 milligrams, which according to the literature and look up into the textbooks, most mated queens weigh about 200 milligrams. So the queens that we were purchasing weren't on the heavy side, they weren't on the light side, they seemed to be just right. There was a lot in any normal bell-shaped curve. There were some that were heavier than others, some that were lighter than others. And we did find that about 21%, or about a fifth of the queens, weighed less than 175 milligrams. I don't know if that's good or bad, but that's what we found. Another aspect of the physical quality of queens was looking at the ovarials. So, you know, one could argue that a queen bee is nothing more than an egg factory, right? Her sole job within the colony is to lay eggs and for the workers then to raise those eggs into new bees. And so her egg-laying machinery, or the ovaries, are very, very important to that end. And so what we've done in collaboration with Dr. Susan Farbach, who's down the road in North Carolina at Wake Forest University and one of the nation's premier neurobiologists and neuroanatomists, is that we dissected out each queen's ovaries, and of course there's 2 of them, and embedded them in some paraffin, and then took cross-section slices of those ovaries in order to determine the number of ovarioles. So each ovary is really kind of a bundled packet of individual kind of spaghetti-like string tubes that you can kind of make out here in this picture of an ovary. And each one of those tubes, known as an ovariole, is its own individual egg production factory where it descends down from her kind of thorax end down to the sting chamber end as the egg matures, as it goes down and gets ready to be laid. So the idea is that the more ovarioles, the more eggs a queen can lay per unit time and therefore be more productive. And while we haven't quite yet finished a lot of this research, we do know from the literature that you know, a high-quality queen, or one that is the most fecund and the highest egg-laying capabilities, typically her ovaries have 150 to 180 ovarioles per ovary. So overall, she has a good 300 plus ovarioles. And what we found, and this is very optimistic, is that our preliminary results suggest that not even 8%, about 7.5% of the queens that we found had fewer than 250 ovarioles, and that the average was over 300. And so there definitely does seem to be that the queens that we purchased had sufficient ovarioles and therefore very functional ovaries. One final thing that we measured as far as kind of looking at the queen's egg-laying capacity was not just looking at the egg factories in the ovarioles, but looking at the main protein that is incorporated into eggs. That protein, many of you who may have heard in some of the beekeeping literature, because it's being discussed more and more, this important protein known as vitellogenin, or VG. And this is the protein that is in the fat bodies of the queen, and it's floating around in her hemolymph, and it gets sequestered and gets kind of taken up into the egg yolk of the egg. So as the eggs mature and they move down the ovaries, they take up this really, really important protein, and sort of the more protein of vitellogenin that a queen has, presumably she'll be able to lay more eggs or higher quality eggs. And so what we did is we measured this this important protein, and this is on a log scale, so each tick here is a factor of 10. And what we found was that it was very highly significantly correlated with weight. So that the heavier that queens were, they tended to produce much more of this vitellogenin, which therefore bodes or is indicative that heavier queens are actually perhaps better or able to lay more eggs, which again may be very intuitive. We also, I didn't mention this before, but some of the queens, after we ordered them, we put them into 5-frame nukes and just let them start laying eggs and start making brood. Other queens we just put in a queen bank and waited for those other queens to lay for a full brood cycle, and that's when we sampled them and measured them for everything. And so by looking at the difference between those two, queens that were actively laying had significantly higher just kind of looking at them and ranking them, whether they were kind of, small, medium, or large kind of thing. And so it may not be kind of comparable to compare apples and oranges here, but at least it gives some reference to historical records of the ovary development of queens. Now, one other, as I mentioned before, one other very important aspect of a queen's physical quality is not just her size or her egg-laying capacity, but also the degree to which she's parasitized. So the main parasites that infect queen bees, it's not Varroa, but rather Nosema, shown here, which is that internal gut parasite, microscopic, and then also tracheal mites, which live in the breathing tubes of the queens and has been problematic for queens and their quality. So in the past, many studies have looked at the degree of parasitism of both Nosema and tracheal mites. Of course, tracheal mites weren't introduced until the mid-'80s, so these earlier studies, it's not really applicable, but studies since then have really looked at the tracheal mites and found fairly significant levels, 31%, 21%, 20% as recently as 1998. Nosema, of course, has been a problem for much longer than that, and in fact, back in the 40s, 50s, and 60s, it was a real major concern of queen breeders, and they were finding, 11, 8, 7% of the queens that were purchased had Nosema in their hind guts at one level or another. So the question is, what are these levels today? Are they higher? Are they lower? And can those levels perhaps explain some of the problems that beekeepers have been experiencing with their queens? So I won't bore you with the details. Many of you probably know about how to actually assay these different parasites, but I'll just cut to the chase. In looking for Nosema in the hind guts of these queens, we found none. And in fact, in dissecting out the hind guts and looking under the microscope for these spores and not having found any, I didn't believe it. It must be, well, we just don't know what a spore looks like or something, and we must not be doing this right because we're expecting, something around 11%. So we, in addition to looking at the spores and finding none, we performed genetic analyses on them that would detect spore levels that may be too small to actually readily detect under a microscope, and we still found none. So of all of the queens that we purchased and assayed, not one of them had Nosema. Similarly, of all the queens we found, only 2.5% of them had tracheal mites, or at least evidence of tracheal mite damage. So this was 3 queens out of the 130 or whatever queens that we looked at. All 3 of these queens came from the same commercial source. So that means that 11 out of 12 of the commercial operations sent us queens that were free of tracheal mites. And again, compared to when they first arrived in the 1980s and later, something almost a third of all the queens that were purchased back then had tracheal mites. I think that this bodes very, very well for the job that a lot of these commercial queen producers are doing. However, one thing, or another entire set of parasites that hasn't been looked at in queens before, but something that's making a lot of headlines, particularly with these new kind of genetic techniques, is to look at virus. And so we know that there are many different viruses out there that infect honeybees, and queens are no different. And so we screen for all of these different viruses, listed down in order from top to bottom here: acute bee paralysis virus, black queen cell virus, chronic bee paralysis virus, deformed wing virus, which everybody knows, of course, is the calling card of Varroa, those shriveled up wings that are very, very cryptic and problematic for a colony. Israeli acute paralysis virus, which has been linked in some studies to potentially to CCD as a marker or possibly even a cause. Its kind of virus cousin, Kashmir bee virus, and then finally sac brood virus. And so what we found among all these queens is that many of them were not infected. So that's what these columns here are. These are absent queens that had no virus at all. But many of them did. And so all of these here are different orders of magnitude, again, in an increasing logarithmic scale, so that some queens had very, very high levels of virus in their tissues. However, there was no real consistent pattern across the different queen breeders, even within a set of queens, just because a queen from the same breeder had a virus doesn't mean that another queen from that same breeder had that same virus or at that same level. So it was really spotty, and there was no real clear pattern to it, other than the fact that we found all of these viruses just as we would if we screened pretty much any of our colonies out there. So in looking at the kind of physical health, or the degree of parasitism of queens that we're purchasing today, I think there's some very good news here in the sense that the two traditional major parasites of queens are at fairly low levels, or at least in this batch of queens that we purchased, but we tried to make it a representative cross sample of what anybody else would buy, and so I think this bodes very well. Okay, finally getting to the mating aspects of queens. So as I said before, there are really two ways to measure a queen's mating health, and that is to look at the sperm that's stored in her spermatheca, and that she keeps there for her entire egg-laying lifetime, as well as the number of different mates represented in her stored sperm, and therefore are the fathers of the worker offspring of her resultant colony. And so traditionally, a lot of studies have looked at the contents of the spermatheca and counted up the number of sperm stored therein. The way that this works is that the spermatheca in the queen's abdomen is this spherical shaped organ, and it has this tracheal net, that's this kind of whitish coating around that tracheal net that bathes the sperm in oxygen so that they can remain there dormant for years if necessary, so that she can then continue laying eggs. That spermatheca is then disrupted into solution, which is this cloudy material in here, and then once it's in solution, it can be, the sperm can be stained and then visualized with a microscope, and therefore counted very accurately. Now, many of you may have seen these fairly famous pictures by Sue Kobe, where the spermatheca of a virgin queen or a poorly mated queen is filled with fluid, but it's very clear. So that spermathecal fluid, you can see right through it, and you know that that's a virgin queen. A fully mated queen has a very full spermatheca of sperm, and it has this kind of tanned, marble-ish look to it, kind of like some fancy bowling ball or something like that. And then something in between where a queen either isn't fully inseminated or she's started to run out of sperm. It takes on this kind of opaquish white color to it. But again, by disrupting it into solution and staining the sperm using these various techniques, you can actually visualize the sperm under a microscope. And so that's one thing that Debbie Delaney really, a technique that she brought into our lab, where you put the sperm under a microscope and you stain it with these solutions, and these little dots that look like you know, the nighttime sky, each one of those is the nucleus to an individual sperm. And so by simply counting up the number of dots in a particular area, you can then assess how many sperm the queen had in the spermatheca. And the literature tells us that a well-mated queen has about anywhere between 5 to 7 million sperm stored in her spermatheca. So that just kind of gives us a benchmark to what we might expect.
This sort of puts counting Varroa in perspective, so I don't think anyone ought to bellyache about counting a few Varroa on a sticky board when you're talking about 5 million sperm.
Well, luckily, unfortunately, otherwise we'd still be doing this.
Still be there on the first slide, huh?
Yeah, we count, for example, this representative field here, and then we can extrapolate out to assess, based on what the total number would be if we did count it. But yeah, it still does cause blindness, and that's why many of those undergraduates are no longer working for us. So again, just to put this into some historical perspective, other researchers in the past have looked at this, at least the sperm number, the stored sperm number of queens, and historically they kind of have this cutoff of 3 million sperm, and anything below that they call that poorly mated. So we'll just kind of use that same cutoff, looking at less than 3 million sperm as being poorly mated. And again, anywhere from 5 to 7 million sperm is a fully mated queen. So a study back in 1962, 30%, 29% of the queens were poorly mated or had fewer than 3 million sperm. 1984, there was a study done where 11%. Most recently, a study by Kamazine et al. At Penn State, they found that about 19% of the queens had fewer than 3 million sperm. So the question is, what are we finding today? Is it possible that the queens that we're purchasing have, on average, lower sperm counts than perhaps they ought? So what we found in looking at the spermatheca, contrary to that prediction, is that the average sperm count of these queens was spot on at about 4 million. So there definitely didn't seem to be drastic decreases in the sperm counts of these commercially produced queens. Using that same cutoff of 3 million sperm, we found that 19% of the queens were indeed poorly mated, which just happens to be the exact same number that was found about 10 years ago. To use a separate cutoff and something that we'll call inadequately mated, that is queens that had fewer than 5 million sperm, we found that 4 out of 5 queens didn't quite reach that 5 million mark. So so I don't know if that's fair to use that as kind of a second bar to reach, but if a well-mated queen has 5 million or more sperm, then only 19% of those queens were above that. So a lot of these queens were obviously somewhere in the middle. They were between 3 and 5 million sperm, which I don't know if that's a good or bad thing, but that's what we found.
Do you have any information that?
Sorry.
I'm sorry. Do you have any information that might shed a little light as to why those 81% were less than 5 million? I mean, what? And maybe you're going to get to this a little later on, in which case just tell me that, and I'll be quiet. But I'm wondering if you have any information about why that such a high number are poorly or inadequately mated.
Yeah, right. Yeah, I think to put out that distinction between poorly mated versus that inadequately mated, I think is important. But it certainly begs that question, doesn't it, Shane? I mean, I'm curious myself. In fact, I won't be presenting any answers to that here.
Okay.
But you're absolutely right that is the next question that we're working on. In fact, we're going to be looking at exactly why, not looking at the number of sperm, but the viability of the sperm and of the queens and the drones. And so that's where a lot of our research is going now. So you'll just have to invite me back some other time so I can talk about that as well.
That won't be a problem at all.
Okay, sounds good. One nice thing about having these large data sets and measuring so many things about queens is that you can then look at the relationships among them all. And so one thing that popped out in our data set was looking at the thorax width and the right wing length of our queens. In other words, body size, not weight, but body size, tended to be very highly correlated with stored sperm count. In other words, so that larger queens, not necessarily heavier queens, but the width, not the weight of the queens, tended on average to have more stored sperm. So if one wishes perhaps to have a proxy for a well-mated queen, or I should say a well-inseminated queen, one can perhaps screen these queens without the use of dissecting them open and therefore killing them and counting their sperm. One could perhaps just look at how wide their thorax is and gauge whether or not she is going to tend to have a higher sperm count than others. And as I said before, there was really no significant effects of the number of viruses or the level of viruses among these queens when it came to her insemination. So how greatly she was infected by these viruses really didn't have any bearing on whether or not she had a large sperm count.
And Dave, did you? This is a question coming in from Larry, actually, who was with us last week talking about splits, and he wants to know: did you measure the diameter of the spermatheca as related to whether it's got more sperm or not?
Is that Larry Connor?
The questioner shall remain anonymous to preserve the innocent.
Well, that person is definitely calling me out. You can tell Larry Doe, anonymous Larry, that is something that I'm still kicking myself for not measuring the diameter of the spermatheca among these queens. But I'll let it be known that we repeated the same experimental design and did measure the diameter of the spermatheca and calculated the volume of the spermatheca, and that is exactly why larger queens have more stored sperm. It's because larger queens also have larger spermathecae, and those larger spermathecae naturally can hold a greater number of sperm, and so that's probably exactly what it is. So that is very insightful, very intuitive, and points to something that I really wish we would have measured in these queens, but it certainly exists.
The anonymous questioner just stated that you have made his day. And at risk of going off on a slight tangent, but we're talking about these larger queens and stuff, and so I'm really going to perhaps throw you to the wolves here, maybe, and you can duck and weave as you see fit. I'm sitting here thinking, okay, larger queens, they've got more VG, potentially more egg production capabilities. You've got larger queens, therefore the spermatheca is larger, if I'm understanding you correctly. Potential to hold more sperm. I find myself wondering if in your sampling of queens from across the country, if you included any small cell queens and how that may play a role in small cell beekeeping that really has sort of gained quite a bit of foothold in this country.
Well, that's a real excellent question, Shane, and I think that there's many layers to this onion and different studies that I'd really like to do. Just because of logistical constraints, we chose to just look at Italian stock and of kind of standard, rearing practices. While they can vary tremendously, none of them were small cell.
Okay.
I don't quite know what the effect of small cell would be on queen cells, since they're different orientation and different sizes. That's actually an intriguing question that maybe you or somebody in the audience knows the answer to, but I don't know if queens raised in small cell colonies are also smaller, or if it's just the workers.
Well, to be honest,
more importantly, sorry, go ahead.
I'm not 100% sure on the answer to that question, but my understanding is, to be honest, I've never tried a small cell colony, but my understanding is, of course, the cells are smaller, and thus you need a smaller queen because sort of quote unquote
Well,
a normal sized
yeah.
queen, her abdomen won't fit in those smaller cells.
Mm-hmm. Mm-hmm. Yeah, no, I think that's an interesting question, an empirical one. I'm not going to be presenting it here, although I had the slides in and I got rid of them probably because of time constraints, and now I wish I didn't. But we did a study where we purposefully raised not queens in small cell, but we raised queens where we grafted older larvae. For those of you that may know some of the intricacies of queen rearing, if you raise a queen from an older worker larva, they are of lower quality because the resultant queens are going to be more worker-like because they've already spent several days in larval development as a worker, so when they transition over into being raised as a queen, they don't make as good a queen. And so we purposefully did that just so that we can kind of see the extremes of the quality from, high quality as good as we could make them, and low quality is pretty much as bad as we could make them, just so that we can kind of see that entire spectrum. And so by looking at that, what we found is, yes, that those purposefully smaller, purposefully lower quality queens had lower sperm counts, but again, largely because they had smaller spermathecae. So yeah, unfortunately I'm not going to be presenting that, but that's kind of a side note that kind of gets to that point. But there's lots of other experiments that would be of interest here.
Okay, thank you.
No problem. Thank you guys for the questions. Keep them coming. This is a lot of fun.
Oh, there's a list developing. Don't you worry.
Okay, good. Well, just to get back to the insemination quality of the queens that we're purchasing today, again using the same historical 3 million cutoff, we're finding that queens are not worse, they're not more poorly inseminated than they have in the past. So, if one of the potential reasons for the queen problems that we're experiencing today, it's not likely to be the number of sperm per se, but as I said before, we're following up on other aspects of that that may indeed lead to poor queen quality. So the last thing that we measured here was the mating frequency of the queens. As you all probably know, queen bees don't just mate with one drone. They mate with multiple drones. And so understanding not just how much sperm they store, but who they mate with and how many times they mate is really quite important. And this is where modern genetic techniques can be very, very powerful. By looking at the worker offspring of queens and doing the genetics on those workers, you can then infer the mating numbers of the queens. I should mention that this has been a measure among these various studies that's never really been done before, in part because, well, it wasn't even known until the 1940s that queens mated more than once, and it wasn't until Norm Gary's work in the mid-60s where it was shown that they mate, about a dozen times. But no study has ever really looked at mating number among the commercially produced queens. So it's a real kind of question mark that they can attain 5 million sperm, but if all of those sperm come from 1 or 2 drones, that may not bode well for colony health. So the way that that studies have done this is taking the queen, she's mated with multiple drones, in this case it's just 2, and then as she fertilizes the eggs fairly randomly from the sperm of these various drones, they create these genetically distinct subfamilies within the colony, and so the colony workforce is therefore highly diverse genetically. And so if one samples these worker offspring and do these molecular tests on them, you can then infer their paternity. In other words, we're trying to find out who their daddies are. You take the workers, you find out, who fathered them, you count up the number of different fathers, and that's how many times the queen mated. So it's a lot of work, but it does give a very robust estimate of the mating number of these queens. Previous estimates, if you just compile all the studies that have done this kind of thing, the average mating number of queens is about 13. So about a dozen drones that queens mate with. However, there is a lot of variation among queens. So some queens do only mate once or twice, or not even 5 times. Other queens have been estimated to mate with upwards of 45 different drones. So there's a lot of range here, and so trying to understand what commercial queens mate with may shed some light on this. And so what we found doing all these paternity tests on these various queens, we found that the queens mated with an average of 15 drones. Now, when a queen mates with that many drones, and she stores a proportion of each drone's sperm in that spermatheca, when she fertilizes the egg, by and large, she fertilizes the eggs randomly. So any given egg can be sired by any particular drone. There's no order or clumping or anything like that as the queen lays the eggs. However, there can be differences in the proportions of the drone fathers. In other words, if a queen mates only twice and you look at 100 of her worker offspring, if 50 of them were sired by one drone and 50 of them were sired by another, then clearly she's effectively mated twice. But if 99 of the offspring are sired by one drone and only one is sired by another, then effectively that queen only mates once, or has only mated once. So there's this kind of weighted average of the effective paternity frequency among the workers. And so taking that into account, we found that that effective mating frequency of queens was about 12. Exactly spot on of what queens are quote-unquote supposed to mate with. So it seems pretty clear that not only are queens getting 4 million sperm and storing 4 million sperm, but they're also mating with an expected number of drones. And so that they're not under-mating or being under-inseminated in most cases. And just for the sake of completeness, looking at, again, the thorax width of the queens with the effective mating frequency, we find this positive relationship, so that larger queens do indeed mate with a greater number of drones, and they store more sperm, and they produce higher levels of vitellogenin. So size does definitely, not necessarily weight, but size definitely may be a good proxy for queen quality. We've also found this correlation here that, again, may not be all that surprising, that as mating number increases, the sperm count increases logistically. So that a queen, to make sure that she gets a good 5 million sperm, she needs to mate with about 12 or more drones. So this is where we stand, and this is pretty much where I'm going to leave it. As I mentioned briefly, that we're going to be continuing in different areas of research along these lines. But I think this is really important for us beekeepers to know that there's a lot of good news in these samples, that contrary to my kind of hunch and very non-scientific, preconceived notion, I went into this really thinking that, well, a lot of the queens that we're getting, they're being superseded early, they're running out of sperm early, therefore they must not be mating well enough. And so I was fully expecting to find this number to be much lower and this number to be much higher. But in fact, what we found is that historically the queens that we're buying today are not really all that different from those that have been produced in the past. And in some cases, like in the case of their parasitism, queen breeders seem to be doing a very, very good job in keeping their queens healthy and well mated. So I think there's some encouraging news here, but it does beg the question very predominantly, well, if they're not poorly mated and the queen quality overall is fairly high, why are we still experiencing these problems with our queens? And so that's where our research is going to be going. We're going to be looking both upstream from the mating process, that is looking at the drones themselves, and that maybe there's something wrong with the drones, that the queens are being adequately inseminated, but the drones are duds, and therefore that's what makes the queens not last as long. We're also looking downstream that, well, maybe, you know, all of these queens were newly mated by the time we bought them. Maybe putting them into colonies for a month or two, something happens that then diminishes their quality, and that's why they don't last as long. So that's where our research is going, and I'll be happy to entertain any questions that you might have.
We do have some questions that people have posed, Dave, but I'd like to start with just someone that threw in when we were talking about small cell. This person raised 6 small cell queens this year, and they did appear to be visibly smaller. While they acknowledged they didn't go through the lengths that you went through to actually measure them, they did at least appear to be so. And these were produced in, I guess, his own operation, not from commercial queen cups. The bees themselves raised them out. So that's just an interesting observation from someone. We have a question sort of about, I guess, a little bit about your design, study design, and they're curious about what time of year did you purchase these queens, and I'll elaborate on that by asking sort of a follow-up and asking, do you think you might see different results if you purchase them at different times of the year?
Oh boy, absolutely, spot on. Excellent question. Something else that we're just kind of dying to look at, and something that 1998 study by Scott Kamazine and others did look at, in fact, and that is the difference between spring queens and fall queens. What we did is we ordered them in the winter, just like any of us have to now, because, you know, these darn orders get filled up so quickly.
Sure.
You know, we ordered them through the winter for a springtime delivery. Around here, that was April and May, April and early May. You know, it would really be nice to add a seasonal component and do the same kind of thing, so looking at spring queens versus those produced in the summer and in the fall. And anecdotally, what the Kamazine et al. Study found was that those in the spring had higher sperm counts than those produced in the fall, which suggests that the queens that are produced later in the season, when there perhaps are fewer drones out there, you know, the conditions may not be as optimal for the mating process, that indeed they're not mating quite as well. We didn't do that as part of this, but hopefully we'll get to that same type of question. But absolutely, it's an excellent question.
This is from our friend Larry again. And I believe this is the work that I often refer to, that I heard you present at SNEBA a few years ago, where you looked at, I believe they were, maybe you grafted these larvae, but basically alluded to super or emergency cells and the age of those larvae and the quality of the queen thereafter. And the question is, at what age larva did you see a significant drop in the quality? So my understanding is sort of the optimal age is right at about day 2 of that larvae's life. Is that not correct? And at what time did you see sort of drop in the quality of that queen thereafter?
Well, the optimal age to raise the best possible queen is the moment that the egg chorion dissolves.
Oh, okay.
So the youngest possible larva makes the best possible queen.
Okay.
So that it's, you know, it's fed royal jelly for its entire larval development. Now you can raise, you know, fairly good queens from 6-hour-old larvae, 12-hour-old larvae. There is a fairly linear drop-off when it comes to the quality of queens. but it's kind of hard to detect statistically for queens that are less than a day old. So it's something that keeps going down the older the larvae get. So by day 2, the quality of queens is substantially different. The absolute cutoff, that is, it's impossible to raise a queen from a worker larva that is 3.5 days old or older. So that's kind of that critical juncture from which the cast is determined. It's either going to become a worker or a queen at that point. It's no longer kind of plastic or flexible. So to make really, really low quality queens, you graft 3-day-old larvae, and they're very, very worker-like, and they're of very low quality. You guys convinced me, of course, that what I need to do is pull up that data that I got rid of, and I will see if I can present that to you. But go ahead and ask another question.
One question is, this sort of gets to the poorly mated, inadequately mated issue a little bit. The question is actually how many sperm, essentially, is a queen going to need for a calendar year? I guess as I'm thinking through, I'm trying to do the math here. You hear the number, a queen can lay about 1,500 eggs a day, and I'm thinking about the average egg-laying season, and there's no way a queen's going to need all those sperm, and so I guess that I'd like to pose that as well. Why do these queens need so much sperm to be considered adequately mated when they only need a small little fraction of it?
Yeah. Well, the short answer is read Larry Connor's book.
He's going to like that plug.
He goes through a lot of the math there and that kind of thing. So that's the short answer. The long answer is that when a queen fertilizes an egg, she releases, and this is some really nice work done in the 70s by John Harbaugh, the former USDA researcher, where the queen releases between 3 and 20 sperm. And then one of those ends up actually fertilizing the egg. But you have to keep in mind that every time a queen lays an egg, she doesn't just use one sperm. She'll release more than one just to make sure that it's fertilized. And so, as she then depletes her sperm source, it can actually be faster than the number of eggs that she actually lays. So just to get back to that previous point, this is some data from the study that I was talking to before about raising high-quality queens from 0-hour-old larvae, and then low-quality queens from I believe in this case it was 48-hour-old larvae. So we wanted to really maximize the kind of queen quality spectrum, and we measured all of these same things: the weight, thorax width, head width, spermathecal diameter in this particular study, the number of sperm, the percent fertility, that is the viability of the sperm, and then the mating numbers of the queens, and found again that all of these things were very, very highly correlated. And so from that, we took all of these measures of queen quality and kind of merged them into one single variable that we'll just call quality. So it's kind of a statistical way to take these multi-dimensional aspects and kind of fuse them into one singular thing. And so that's what this continuum here is, from very low quality to very high quality. And what we found was that queens that were grafted from young larvae were very high in their quality, and queens that were grafted from older larvae were very low in their quality, very highly significant difference. So that kind of generated the bounds of reproductive quality of these commercial queens. So, we purposely made really, really bad queens, and then ask the question, well, where do the commercial queens fit along this spectrum? So if this was an A plus and this was an F, can you guys see the cursor as I'm doing this?
I can, yes.
Okay, sorry. So if the very left-hand side is an F, this is an A plus, what we found for these commercial sources is that they were on the right side of the middle, that they got a final grade of 79% or C plus. So again, I think that there are there's very good news in this, in the sense that these queens are definitely above average. It's also good news in the sense that there may be things that we can improve as far as queen quality among these commercial queens. And one thing that we're trying to work with some commercial queen breeders, particularly those out in California, is hopefully come up with some very simple rules and methodologies to kind of get rid of this left-hand tail. So if we can candle queens, or if we can measure queens, and then kind of cull the lower quality ones before they go out and mate and before they're actually sold, then we'll be able to improve the overall average queen quality and therefore help improve the product that we're all buying. So this was the slides that I got rid of, and I now
they're back.
regret that I did, but there you go. Benefits of having the computer right here.
It is nice, isn't it? There's another question about one of your parameters. Why did you measure the thorax as opposed to the abdomen?
Yeah, that's a good question. The reason being that it's traditionally been a lot more reliable in the sense, in two very important ways. One is that the thorax is much less flexible to changes in a queen's weight. In other words, when a queen is a virgin and her ovaries aren't fully developed, she has a very slender and very smallish abdomen, and that's so that she can fly and that she's not going to be burdened down. When she has very developed ovaries, thorax width tends to pop up a lot more readily than things like weight, or if we did measure it, abdomen width.
I actually lost you there for a second. I'm having some technical difficulties up here. I'm at a place that I'm not normally out of, and every time I get a fax, I lose my internet connection.
Oh dear.
Yeah. So sorry about that if I've sort of seemed to have dozed off there. It's not that I've dozed off. I lost you. There's a couple questions here, sort of asking about. Obviously, you only looked at Italian queens, but this is more an opinionated type question about Russian bees or any other type of queen, yeah. Buckfast, Carniolan, etc. And I realize that being a researcher, you sort of try and let data speak and be sort of objective, but I'm wondering if you might venture out a little bit there.
Well, it would be something that I would like to do,
mm-hmm.
but my overwhelming hunch is that there wouldn't be really any differences among the stocks, if you, or at least you wouldn't really be able to detect any real substantial difference just based on the stock. I think what drives most of the variation in mating number is not, I mean, well, the queen's behavior and her genotype and everything may have an impact. I think the main impact is the local mating conditions, that is weather and, more importantly, the drone population. So that you can take, you know, a Russian queen and an Italian queen, mate them in the same place, they're probably going to mate with the same number of drones. You put them in the middle of the desert where there's hardly any, you know, colonies, they're going to mate fewer, but they're probably going to mate with the same kind of correspondingly fewer drones. So that's just my hunch. I believe that there are some other researchers that are looking into this. I certainly know that other studies that have looked at different races of bees, that is bees in their native Africa and Europe, looking at different areas that there are differences, but they're not drastic differences, and it's more likely due to the mating environments more than the stocks themselves.
The previous webinar that we did with Larry, he showed some very interesting slides from a banked queen versus a non-banked queen, and I'm wondering how you think duration of banking may play a role in some of these factors, or just banking in general, how that may play a role in some of these parameters that you were looking at.
Well, I'm starting to sound like a broken record saying that, yeah, we're going to look into that, you know, but we really are, because that absolutely is a very crucial aspect to kind of the queen's reproductive capacity, that upon mating, the queen shuts down her behavior to mate, and she turns on all of her egg-laying machinery. She starts, you know, laying eggs and getting that up and going. And then what happens? Some beekeeper takes her, slaps her in a cage, and sends her through the U.S. mail for 4 days. So she kind of shuts down her queen rearing again. Or they go and put her in a bank, and therefore not exposed to comb. Her ovaries, the eggs stop developing. Once she's put back into a colony and has to start all of that egg-laying machinery all over again, it's been my experience, at least, that sometimes they pop the clutch. And I don't know what Larry was saying last week, but it's very possible. I think it's important to understand the physiological processes and the effects that shutting down a queen's egg-laying has on her potential future egg-laying, and how often you know, it doesn't kick back in again. I think that that's definitely something that should be looked into and could have some real important insights into what we do.
Okay. Here's a question just of curiosity from someone, is in terms of how far queens and drones fly to mate.
Yeah, that's a good question. Pass. No. The answer is better addressed by Chip Taylor. Chip Taylor is you know, a fairly renowned honeybee researcher, and I had the privilege of joining him and Marla Spivak and others in Northern California looking at drone congregation areas in these commercial mating yards. And he has this uncanny ability and sense to detect where mating is actually going to take place. And contrary to what we would like to have is some sort of hard and fast rule that queens always fly 1 mile to the south-southeast, and that's where mating takes place. That's not how it works in these mating systems. It's difficult to predict. It's even more difficult to find, but somehow or another, all of the queens and the drones in a particular area know to meet up in a certain area, very high off the ground. 30, 50 feet up in the air, and that's where mating takes place. So it really varies tremendously based on location, and there's real no good, hard and fast landmarks or anything that the bees use in order to make that determination, but somehow they're able to figure it out.
Okay, I have a question, and I'm not going to put you too much on the spot here by asking, of course, the breeders and things like that. But you mentioned right at the beginning that you purchased from small and large producers, and I'm wondering if you sort of isolated those two at all, just based on size in your study, to say, okay. Generally, people that are producing fewer queens, the quality tended to be a little better or worse. If you saw anything like that, or if that's something that you can glean by going back into the data.
Well, that is what we're doing, not on this particular data set, but on our subsequent data set, on a larger sample that we've run since then. Looking at the number of queens produced, looking how many mating nukes they have, how many drone source colonies they have, what chemical treatments they're using to control varroa mite and nosema and all these other things. So we're going in and actually working with those queen producers because they're dying to know too. They really, it's been very open and welcoming because they really want to improve their product as well. And so we're working hand in hand with them to try to see what management practices they're doing has an end effect on the queen quality. Not ready right now to say anything definitive because we still need to crunch the numbers a bit more. But that's the ultimate goal, is to try to see what management practices are being conducted by these queen producers, large or small, that could help them cull that lesser, that low quality tail and really help improve the overall product that they're making.
Okay. I guess I have just one statement that I'd like to make on your behalf, David. And one of the criticisms that I've heard from people, beekeepers, about research very much like this, or perhaps on a similar topic or of nature, that criticizes the researchers to say, okay, well, they've got all this great data. Why don't they tell us who the best producers are? And I just want to say, I'm not looking for you to respond to this, but I think that, from my perspective, of course, I would love to have the answer to that question, but I understand completely your reasons for not divulging that information, that I think it's important as a researcher to remain objective. And I think if you were to assign identity to those letters that were across the bottom of that graph, you lose some credibility as an objective researcher, I think. And that's not so much a question as it is a statement, because I think researchers sometimes take a lot of heat, and frankly, I can understand that from the beekeeper's perspective, but I think it's important to sort of put ourselves in your shoes for a minute and understand why that information is not being divulged. So.
Well, I also think, Shane, that you make a good point that, in working with these producers, I think anonymity is really important, because we certainly don't want anybody to be shown to be, producing bad queens, and that could adversely affect their business, or that they turn around and they use it as a promotional campaign, and use that. Science should not be about that in the marketplace. So that, your point is well taken. But I would also submit to you that in this particular case, it doesn't matter. And this is why. Right. We were talking before about all these various things that affect the reproductive quality of queens. And we said that, spring queens versus fall queens, anecdotally, there's big differences there. We looked at all of these different queen producers, and there were huge differences within each producer, and which swamped out the differences among them. And I guarantee that if we would go and buy, another batch of queens the following year from each of these same producers, the rankings of who made the best queen and who made the worst queens would be not consistent with the previous year. Does that make sense?
Sure,
In
yeah.
fact, we have done this so that, you know, now if there were huge differences among them because they're doing, you know, very, very different things, then that would be another story. But because we're not seeing but subtle differences among the producers, even though they are significant, they're still subtle, and I don't think they're consistent. I think they deal with other factors that, like weather and other things like that, that are not necessarily in one's control. So I would submit, and I do this all the time when I see the audience out there and I ask for a show of hands, you know, because that's the #1 thing everybody wants to know. Who's producer A and who's producer G, you know?
Sure.
And as I said, not only the anonymity in working with this to help improve the product, but also, you know, I think that it's irrelevant. That really the question is, what do we expect to get on average whenever we go out and buy queens as a total population?
Right.
You know, what are we expecting to get? And it comes down to, if you find a good queen producer that you're happy with their queens, keep using them. If you get a bad batch of queens from a queen producer, go to somebody else, or, you know, work with that producer and say, hey, you know, I didn't quite like those queens. Can I get another set? Or, you know, I think that it really comes down to exercising our, you know, power of the pocketbook as consumers of honeybee queens as a product. And so that's why I think also that the coded queen producers is really necessary, because knowing who they are in many ways is irrelevant.
Okay. I've just got one sort of, it's a two-parted closing question here, because we're approaching now 7:30, and you've been very generous with your time. The information that you presented tonight was excellent and very informative. But as I was going through it, I was thinking, okay, well, a beekeeper is not going to be able to call up a breeder and say, well, what's the diameter of your, on average, your queen spermatheca, or how many sperm are in your mated queens, and things like that. So what sort of things, not yet, you're working on those tests, huh?
Well, I mean, exactly. I think, you know, working with the producer, because they would love to have that information, or at least have a correlate to that. So if they could use thorax width or wing length or something like that as a proxy for some of these important measures, and if we can establish that into the culture of queen producers, I do think that might be useful for the consumer, and that they would pay attention to that. I think it's too early right now to really see if that's necessary, but I think that is one potential outcome of a lot of this that would empower the consumer by being able to shop around and have some more objective criteria about the queens that we're purchasing.
So did you, the data that you collected, did you give that back to the producers and say, oh, by the way, we did this sort of anonymously, and here are the results, not necessarily let them know what letter they were so they could figure out sort of where they stood against others, but did you send that back to the producers, just out of curiosity?
I did in some cases, but I did not divulge what the other producers did.
Okay.
Certainly their names.
Yeah, sure.
Just the mean of their queens, and you know what we found, and then the global mean.
Okay.
So we divulged that information. I think as this starts to come out, you know, and again, as we're working with these individual producers, we are divulging it, but again, staying more at a global scale rather than the, you know, us versus them, you know, type of comparison, because again, I think that in many ways that's irrelevant. But in the end, what I would really like to see is, and it might be kind of too far off in the future right now, but I think it would be great to have some sort of diagnostic service that queen producers could send batches of their queens or a subset of every, grafting round that they do or something to, to test the queens and to verify, what kind of quality they are. And, if they make a bad batch, they can just scrap that, or there would be some sort of service that queen producers could use to try to, maintain the quality of their product. Because as I said before, they're very interested in upholding that high standard. And so it would be nice for them to have resources to go to do that. But again, that's far off in the future at this point.
Right. So until we get to that point, what things should a beekeeper that's looking to purchase queens, what things should they be asking their queen producers? What things should they be looking for when they receive these queens to do sort of a quick and dirty assessment of, yeah, she's going to be okay or maybe not?
Well, I think, and again, this is alluding to a lot of the work that Marla Spivak has been doing with the California queen breeders. One is to ask them about their stock selection policy. What are the traits that they're specifically breeding for? What are they looking for in their stock? And does that correspond to your interest as a beekeeper? Are they specifically looking for honey production, which is your interest, or is your primary interest varroa resistance and hygienic behavior? So ask them about what they do to kind of uphold the genetic integrity and quality of their particular stock. If they kind of, hem and haw or don't really have much of a plan, they just say they take the best one and that's it, that you might want to go shopping elsewhere. A second one, and this deals with the mating quality of the queens, and that is ask them about the drone source colonies. How many drone sources do they put out for, how many mating nukes? What types of things do they do as far as getting their queens adequately mated? And then the third thing that I'd ask is how long the queens tend to stay in the mating nukes laying before they're put into a cage and mailed and shipped off.
And what sort of answers, I'm sorry, but what sort of answers would you like to hear to those questions? So they ask these questions. Now, what if they say, okay, well, they're in the mating nuke for 5 days, and I've got 2 drone colonies per 60 mating nukes?
Well, the laying period of the queens, there's been some research out of Australia, which I don't think it's really been followed up on much, but it probably should, that suggests that if a queen is in the mating nukes for a good brood cycle, so that she's laying for a good 3 weeks, that drastically improves the resultant quality of her brood pattern and colony productivity. So in essence, the longer the better is what I'd be looking for there. And much like in the number of drone source colonies, typically the more the better. That that if they don't really seem concerned about how many drone colonies they have, if they give some answer like, oh, we got lots of bees around, there's plenty of drones, that's a good sign that they're not really thinking of the male aspect of the mating process. If they're saying, oh, we put 2 frames of drone foundation in every single one of our strong hives, and we have yards of 2,000 mating nukes, and at each one of those yards we put no less than 150 drone source colonies half a mile away to make sure that the local area is saturated with our drones. That's a good sign, that a very high ratio like that of mating nukes to drone source colonies really shows that they're putting a lot of thought into that. Again, you can't determine, that doesn't necessarily predispose that they're going to mate well, but it shows that they've been through thinking about these types of things, and that's a good sign.
Right.
So there's no guarantees in it, but that's what you want to be looking for as the consumer.
Okay, one last question, I promise. Now we've got a lot of people on there online right now that dabble in queen breeding. They're not a commercial breeder. They're not one that would be in the back of the journals. What if you had to give the most important things? What are the most important things they should consider to produce a good quality queen in a very small backyard operation?
Really excellent question, and a very difficult one too. I believe that in all of us, or the vast majority of us as beekeepers, we all have in us the power to raise our own queens and to kind of, hold our own genetic destiny in our hands. That, that we can do a lot, of doing that. But that said, there's a lot of steps along the way where things can go wrong, right? And for small-time queen rearers to make your own queens, if I had to say that the number one thing to do to make a real high-quality queen is graft them young. If you're going to actually use the Doolittle method of physically transferring worker larvae into queen cups, and then putting those queen cups into queenless colonies so that they'll raise them as queens, make those larvae that are transferred as young as possible. I always like to say that if the larvae are so small that you can't see them, that's the perfect size. If there's a little pool of royal jelly on the bottom, you don't really see the egg casing in there anymore, and there's just only a hint, a glisten of a larva in that pool of royal jelly. That's the perfect size that you want to use because that's going to make the highest quality queen. Again, there's no guarantees. There's lots of other things that are important as well, but I think one of the main things that a lot of first-time or fairly inexperienced people that raise their own queens, they take fairly old and substantial larvae, because they're a lot easier to see, obviously, and graft those, and they don't make as high a quality queen.
All right. Well, with that, Dave, I'd really like to thank you for your time this evening and presenting this information to us and answering all these questions. I know that you've got a very, very busy schedule. I also know that you've got a family waiting for you at home. And so I'd again just really like to extend my appreciation for you doing this for us. You've got some tremendous information, and sharing it with the folks out there is a great asset to us all. And so thank you very much for being with us this evening, and thanks for those that tuned in. We'll keep these going, hopefully. And Dave, when you get some more results on some of these follow-up studies that you kept mentioning this evening, you let me know, and we'll schedule another one of these and get you back online here.
All right, that sounds great. My thanks goes to you and for all of those that stuck with the attendance. I really appreciate this format, and thanks again for the opportunity.
Thank you. Have a good night, everyone.